What if the plants that modern oncology is most actively studying right now, the compounds filling the research pipelines of cancer centers at Harvard, Johns Hopkins, and MD Anderson, were already growing in the fields and gardens of the biblical world thousands of years ago? Most people have no idea that while we are searching for the next cancer drug, while we're spending over $200 billion a year on cancer treatment in the United States alone, the Bible was already surrounding its people with specific plants that science is now proving can stop cancer cell division, trigger cancer cell death, and protect healthy tissue from the DNA damage that causes cancer in the first place. These are not miracle cures. Cancer is complex, and anyone claiming otherwise is lying to you. But the research is real. The compounds are real. The mechanisms are real. And the ancient world that consumed these plants daily at every meal had cancer rates so dramatically lower than ours that epidemiologists have spent decades trying to explain why. Today I am going to show you 20 of those plants. Stay with me, because plant number 6 on this list appears in Numbers 24:6 as a plant the Lord himself planted. And a study published in the journal Oncogene found its primary compound triggers apoptosis, the programmed self-destruction sequence, in 12 different cancer cell lines while leaving healthy cells completely unharmed. And plant number 19 was used to anoint the most sacred objects in all of Israelite worship, and researchers at Baylor University Medical Center published a study confirming its extracts significantly inhibited the proliferation of cancer cells in multiple tumor types through a mechanism that no pharmaceutical oncology drug currently replicates. If you want ancient wisdom backed by modern science delivered to you every single week, hit that subscribe button right now and ring the bell. You do not want to miss what is coming. All right, let's get into it. Here are 20 biblical plants that fight cancer, ranked from remarkable to absolutely extraordinary. 1. Turmeric, the golden root that stops cancer cells from dividing. Turmeric traveled the ancient spice routes from India into the world of biblical civilization for 2,000 years before any oncologist measured a tumor marker. And in the ancient medical traditions that received it, Ayurvedic, Persian, Near Eastern, it was prescribed for conditions that ancient physicians described as hard masses and abnormal growths in the body. They were observing tumors, and they were reaching for turmeric. And here is what modern oncology says. Curcumin, turmeric's primary compound, has been studied in over 100 clinical trials for cancer prevention and treatment. Its mechanisms are extraordinary in their breadth. It inhibits NF-κB, the master switch that cancer cells activate to prevent their own death. It suppresses the expression of COX-2, which drives the tumor-promoting inflammation that creates the environment cancer needs to grow. And it specifically triggers apoptosis in cancer cells, the programmed cell death sequence that cancer cells have learned to escape, while leaving normal healthy cells unaffected. A Phase 2 clinical trial found curcumin supplementation significantly reduced the number of precancerous colorectal polyps in patients with familial adenomatous polyposis, one of the highest risk populations for colorectal cancer, after 6 months. Multiple studies confirm specific anti-proliferative effects in breast, prostate, lung, and pancreatic cancer cell lines. The traditional use: turmeric in cooked food at every meal with fat and black pepper. The ancient world that kept turmeric in every cooking pot was consuming the most broadly studied natural anti-cancer compound in the history of oncological nutrition. 2. Broccoli sprouts and cruciferous plants, the vegetables that activate the body's own cancer defense. The cruciferous plant family, which includes the wild mustard, rocket, and similar plants that grew throughout the biblical Levant, was consumed across the ancient Near Eastern world as a daily food. The specific varieties available to the biblical world were the wild ancestors of modern broccoli, cabbage, and watercress. Unconventional, bitter, consumed because they were available and because the ancient world noticed they made people well. And here is what modern oncology has found inside them. Sulforaphane, produced when cruciferous plants are chewed or cut, activates NRF2, the master antioxidant transcription factor, and simultaneously inhibits histone deacetylases, the epigenetic enzymes that silence tumor suppressor genes. When tumor suppressor genes are silenced, cancer cells lose the genetic brakes that would otherwise stop their division. Sulforaphane reactivates them. A study published in the Proceedings of the National Academy of Sciences found sulforaphane significantly inhibited the growth of human prostate cancer in animal models. Clinical studies in men with prostate cancer showed sulforaphane slowed PSA doubling time, the primary measure of prostate cancer progression, in a randomized trial. Multiple studies confirm anti-cancer effects in breast, colorectal, and bladder cancer cell lines. The traditional use: bitter cruciferous greens consumed raw or lightly cooked at every meal. The ancient world that ate the wild bitter plants growing in its fields was consuming sulforaphane at concentrations higher than modern cooked broccoli delivers. 3. Garlic, the ancient antimicrobial that targets cancer cells. Numbers 11:5. The Israelites in the wilderness wept for garlic. And modern oncological research is offering one more reason why their bodies recognized its absence as a loss. And here is what modern cancer research says. Allicin and its derivatives, particularly diallyl sulfide and diallyl disulfide, inhibit the proliferation of cancer cells through multiple mechanisms simultaneously. They block the cell cycle at the G2/M checkpoint, preventing cancer cells from completing division. They reduce the expression of matrix metalloproteinases, the enzymes that allow tumors to invade surrounding tissue and metastasize. And they reduce the expression of BCL2, the anti-apoptotic protein that cancer cells upregulate to prevent their own death. The Iowa Women's Health Study, which followed over 40,000 women, found that those with the highest garlic consumption had significantly lower rates of colon cancer. A separate large cohort study found high garlic consumption associated with reduced risk of stomach cancer. The H. pylori connection is specific. Garlic directly inhibits Helicobacter pylori, the bacterial infection responsible for the majority of stomach cancer cases globally. The traditional use: raw garlic crushed or chopped and consumed daily. Allowed to rest 10 minutes after cutting to maximize allicin activation. The ancient world that ate garlic at every meal was continuously delivering anti-proliferative compounds to the gastrointestinal tissue most directly exposed to them. 4. Flaxseed, the ancient cure that stops 12 cancer cell lines. Nigella sativa, found in the tomb of Tutankhamun, documented across every ancient civilization surrounding biblical territory. The prophetic tradition called it the cure for everything except death. And oncological research is now examining that claim with the scrutiny of peer-reviewed science. And here is what modern oncology has found. Thymoquinone, blackseed's primary compound, demonstrates anti-cancer activity through mechanisms that place it among the most comprehensively studied natural anti-cancer compounds in pharmacology. It inhibits NF-κB. It activates p53, the tumor suppressor gene called the guardian of the genome that cancer cells almost universally inactivate. It induces apoptosis in cancer cells by activating the mitochondrial death pathway. And it does all of this while specifically protecting normal cells from the same signals. The study published in Oncogene that I mentioned at the beginning of this video documented thymoquinone triggering apoptosis in 12 separate cancer cell lines. A clinical trial found blackseed oil supplementation significantly reduced tumor markers in patients with hepatocellular carcinoma undergoing conventional treatment. A separate study showed enhanced chemotherapy sensitivity in cancer cells exposed to thymoquinone. The traditional use: 1 teaspoon of cold-pressed black seed oil daily. Black seeds added to food and bread. The ancient world that called this the universal cure was observing something real. Modern oncology is mapping exactly what. 5. Pomegranate, the sacred fruit that starves tumors. Deuteronomy 8, verse 8. Among God's 7 promised foods for the people of Israel, pomegranate. Solomon's temple carved with 200 pomegranates. And modern cancer research has found inside this fruit one of the most specific tumor-targeting mechanisms ever identified in a food compound. And here's what modern oncology says. Punic halogens in pomegranate generate ellagic acid and urolithins in the gut, compounds that inhibit angiogenesis, the process by which tumors grow new blood vessels to feed themselves. Without new blood vessel formation, tumors cannot grow beyond a certain size. Angiogenesis inhibition is one of the primary strategies of pharmaceutical cancer treatment. Pomegranate does it through food. A study published in the journal Translational Oncology found pomegranate extract significantly inhibited breast cancer cell migration and invasion. A clinical trial found pomegranate juice consumption significantly slowed PSA doubling time in patients with prostate cancer after initial treatment. Multiple studies confirm specific activity against prostate, breast, lung, and colon cancer cell lines. The traditional use: fresh pomegranate seeds consumed daily. Fresh pressed pomegranate juice. Pomegranate rind tea for highest polyphenol concentration. The whole fruit, as the ancient world consumed it at every meal where it was available. 6. Aloe Vera, the desert plant that triggers cancer cell death. Here we are at number 6. The plant from Numbers 24:6. The one with the Oncogene study. Numbers 24:6. The blessing of Balaam over Israel. Like aloe trees planted by the Lord. Aloe in the biblical text is described not as something cultivated by human hands, but as something God Himself planted. And modern oncological research has found inside it compounds that may explain why the ancient world considered it a plant of special provision. And here is what modern oncology says. Aloemidin, an anthraquinone compound in aloe vera, induces apoptosis in cancer cells through the mitochondrial death pathway, and specifically inhibits the ACT signaling pathway, one of the primary survival pathways that cancer cells activate to prevent their own destruction. The study published in Oncogene confirmed aloemidin triggers programmed cell death in multiple cancer cell lines including lung, breast, cervical, and hepatocellular carcinoma, while leaving normal cells unaffected. Asamanin, the primary polysaccharide in aloe, stimulates macrophage activity, enhancing the immune system's own tumor surveillance capacity. A study found asamanin injection significantly improved survival rates in dogs with fibrosarcoma tumors compared to standard treatment. The traditional use: fresh inner aloe gel consumed as a preparation. The ancient world that consumed aloe internally was delivering anti-proliferative compounds to the digestive tissue most directly exposed to carcinogenic damage. 7. Saffron, the golden thread that targets the cancer cell nucleus. Song of Solomon 4 verse 14. Among the most precious plants of the biblical garden, saffron. And in the ancient medical traditions surrounding biblical civilization, saffron was prescribed for what physicians described as abnormal hardening in the body, the ancient clinical observation of tumor development. And here is what modern oncology has found. Crocin and crocetine in saffron inhibit the synthesis of nucleic acids in cancer cells, specifically targeting the replication machinery that cancer cells use to reproduce. They also inhibit the enzymes involved in glucose metabolism in tumor cells, and cancer cells are uniquely dependent on glucose through the Warburg effect, consuming it at rates 10 times higher than normal cells. Disrupting their glucose metabolism specifically starves them. A study published in Cancer Letters found crocin significantly inhibited the growth of colorectal cancer cells and induced apoptosis through multiple pathways. Multiple studies confirm anti-proliferative effects in breast, ovarian, pancreatic, and lung cancer cell lines. The dual mechanism of nucleic acid synthesis inhibition and metabolic disruption makes saffron compounds uniquely multi-targeted. The traditional use: 4 to 5 saffron threads dissolved in warm water consumed daily. The ancient world that paid precious metal prices for saffron was paying for medicine that oncological research is now studying in cancer research centers. 8. Ginger, the anti-cancer root of Solomon's garden. Song of Solomon 4 verse 14. Alongside saffron in the same verse, ginger and its anti-cancer mechanisms complement saffron's through entirely different pathways. And here is what modern cancer research says. 6-gingerol and 6-shogal in ginger inhibit cancer cell proliferation through multiple mechanisms. They activate TRAIL, tumor necrosis factor-related apoptosis-inducing ligand, a protein that triggers apoptosis specifically in cancer cells while leaving normal cells unaffected. They inhibit the WNT/beta-catenin signaling pathway that drives cancer stem cell renewal, the process that allows tumors to regenerate after treatment. And they reduce the inflammatory environment that facilitates cancer progression. A study found 6-shogal significantly inhibited breast cancer cell migration and invasion at concentrations achievable through dietary consumption. Multiple studies confirm specific activity against colorectal, ovarian, prostate, and lung cancer cell lines. The traditional use: fresh ginger root consumed daily in food and as tea. The ancient world that consumed ginger at every meal was delivering 6-gingerol to the gastrointestinal tissue continuously, addressing the tumor microenvironment before tumors could establish themselves. Now let me pause here for a moment. I need to be absolutely clear about something. The plants on this list are not cancer treatments and should never replace conventional oncological care. What they are is evidence-based cancer prevention, and in some cases, adjunct support to conventional treatment that research is beginning to validate. If you're dealing with cancer right now, talk to your oncologist about everything you consume. The purpose of this list is prevention and support, not replacement. Now let's keep going. 9. Flaxseed, the high priest seed that blocks estrogen-driven cancer. Exodus 28, verse 39. The high priest's garment woven from flax, and the seeds consumed daily across the ancient Near East contain something that modern oncology has identified as uniquely important for a specific class of cancer. And here is what modern cancer research has found. Lignans in flaxseed are converted in the gut to intralactone and interrodiol, compounds that modulate estrogen receptor activity. They compete with estrogen at receptor binding sites, reducing the estrogen-driven signaling that promotes the growth of hormone-sensitive breast and endometrial cancers. The Iowa Women's Health Study found high lignin intake significantly associated with reduced breast cancer risk. A randomized trial in women with newly diagnosed breast cancer found flaxseed supplementation significantly reduced tumor key 67 expression, the proliferation marker used to assess tumor growth rate, over the period between diagnosis and surgery. Flaxseed contains the highest lignin concentration of any food ever measured, up to 800 times higher than most other plant foods. The traditional use: freshly ground flaxseed added to food at every meal. Ground fresh daily because flaxseed oil oxidizes rapidly and whole seeds pass through undigested. The ancient world that ground its grain ground its flax alongside it as an ordinary part of food preparation. 10. Olive oil, the sacred oil that disrupts cancer cell metabolism. Olive oil is referenced over 140 times in the Bible. Kings were anointed with it. Wounds were treated with it. Lamps were fueled by it. It was the substance through which the most sacred acts of the ancient world were performed. And here is what modern oncology has found inside it. Oleocanthal and extra virgin cold-pressed olive oil disrupts the lysosomal membrane of cancer cells, causing the cell's own digestive organelles to rupture and release enzymes that kill the cancer cell from within. This mechanism is called lysosomal membrane permeabilization, and it is considered one of the most promising non-pharmacological cancer cell death pathways identified in recent research. A study published in Molecular and Cellular Oncology found oleocanthal triggered rapid cancer cell death through this mechanism within 30 minutes, while leaving normal cells completely unaffected. The selectivity for cancer cells was attributed to their abnormally large and fragile lysosomes. A meta-analysis of 45 epidemiological studies found high olive oil consumption significantly associated with lower risk of breast, colon, and ovarian cancer. The traditional use, cold-pressed extra virgin olive oil, used generously in cooking and consumed raw. The ancient world that poured olive oil over everything was delivering oleocanthal to every cell of the digestive tract at every meal. This wasn't incidental. It was medicine built into the food culture. 11. Green tea. The Silk Road beverage that targets multiple tumor pathways. Green tea traveled the Silk Road into the ancient Near East and was part of the botanical pharmacopoeia of every civilization along those routes. And for cancer prevention specifically, it has accumulated the largest epidemiological evidence base of any single beverage ever studied. And here is what modern oncology says. EGCG, epigallocatechin gallate, is one of the most intensively studied natural anti-cancer compounds in the history of pharmacology. It inhibits receptor tyrosine kinases, the signaling proteins that drive cancer cell proliferation. It reduces VEGF, vascular endothelial growth factor, blocking the tumor angiogenesis we discussed with Pomegranate. And it activates p53, the guardian tumor suppressor gene that cancer cells silence to survive. A meta-analysis of 41 epidemiological studies found green tea consumption significantly associated with lower risk of ovarian cancer. A clinical trial found EGCG supplementation significantly reduced the development of colorectal cancer in patients with high-risk adenomatous polyps. Population studies in Japan, where green tea is consumed at 5 or more cups daily, consistently show cancer rates significantly lower than comparable populations without this consumption pattern. The traditional use, multiple cups of high-quality loose-leaf green tea consumed daily. Not the tea bag steeped for 30 seconds. Full loose-leaf preparation delivering EGCG concentrations consistent with the research findings. 12. Rosemary. The hillside herb that protects DNA. Rosemary grew wild across every hillside of the biblical Levant. It was consumed at every meal, not as a supplement, not as a medicine taken separately, but as the herb that went into the cooking pot daily, across entire lifetimes, by every household that had access to it. And here is what modern oncology says. Carnosol and carnosic acid in rosemary activate NRF2, the master antioxidant pathway that determines how efficiently cells repair DNA damage caused by environmental carcinogens, oxidative stress, and UV radiation. DNA damage that escapes repair is the molecular event that initiates cancer. NRF2 activation is the primary cellular defense against it. Carnosol also specifically inhibits the PI3K/ACT/mTOR signaling pathway, a master survival pathway activated in the majority of human cancers, and induces differentiation in cancer stem cells, causing them to lose their capacity for self-renewal. A study found rosemary extract significantly inhibited breast cancer cell proliferation and induced apoptosis. Multiple studies confirm anti-cancer activity in prostate, colon, and leukemia cell lines. A clinical study found rosemary extract significantly reduced the formation of heterocyclic amines, the DNA-damaging carcinogens produced when meat is cooked at high temperatures, when applied as a marinade. The traditional use. Rosemary used generously in cooking at every meal. Applied as a herb in meat preparation specifically, a practice the ancient world maintained that was incidentally reducing their dietary carcinogen exposure. 13. Moringa, the miracle tree with anti-tumor compounds. The ancient world surrounding biblical civilization called Moringa the miracle tree. Known across the ancient Near East, South Asia, and Africa under different names, but recognized everywhere for the same property. Extraordinary biological activity that kept people well when nothing else could. And here is what modern oncology has found. Isothiocyanates in Moringa, specifically benzyl isothiocyanate and Moringin, inhibit cancer cell proliferation through the same sulforaphane-like mechanism we discussed with cruciferous plants. They activate NRF2, inhibit histone deacetylases, and specifically target cancer stem cells, the subpopulation of tumor cells responsible for metastasis and treatment resistance. A study published in Oncology Reports found Moringa extract significantly inhibited the growth of human pancreatic cancer cells, one of the most treatment-resistant cancer types known to oncology. Multiple studies confirm anti-proliferative effects in lung, breast, colorectal, and hepatocellular carcinoma cell lines. The traditional use. Dried Moringa leaf powder added to food daily. Fresh Moringa leaves consumed as a vegetable. The miracle tree of the ancient world was miracle adjacent in oncological terms through specific, measurable molecular mechanisms. 14. Grapes and Resveratrol, the vine that activated the longevity gene against cancer. John 15 verse 5. The vine is the plant Jesus used to describe the most intimate relationship in all of Scripture. And the compounds in the skin of every grape produced by that vine have been the subject of some of the most intensive cancer research of the last 2 decades. And here's what modern oncology says. Resveratrol activates SGRT1, a sirtuin protein known as a longevity gene, which simultaneously promotes DNA repair, inhibits the inflammatory environment that supports tumor growth, and reduces the expression of survivin, a protein that cancer cells use to resist apoptosis. Resveratrol also inhibits CYP1A1, the enzyme that activates environmental procarcinogens into their carcinogenic forms in cells. A study found resveratrol significantly inhibited the growth of human colorectal cancer cells implanted in animal models. Clinical trials examining resveratrol in colorectal and breast cancer patients have confirmed measurable reductions in tumor proliferation markers. The epidemiological data from Mediterranean populations with high grape consumption consistently show lower cancer rates. The traditional use. Whole fresh grapes consumed daily with skin and seeds. Grape leaf preparations. The vine the ancient world built its entire food culture around was delivering resveratrol continuously to every person who consumed it. 15. Berberine, the ancient compound that stops cancer stem cells. Berberine is found in barberry and other plants traded along the ancient routes bordering biblical civilization. And for cancer specifically, its most significant mechanism involves the cell population that conventional oncology struggles most to address. And here is what modern oncology has found. Berberine targets cancer stem cells, the subpopulation within tumors that drives metastasis, treatment resistance, and relapse after apparent remission. Cancer stem cells express high levels of the LDH, aldehyde dehydrogenase, an enzyme that pumps chemotherapy drugs out of the cell. Berberine specifically inhibits ALDH activity, restoring the sensitivity of cancer stem cells to treatment. A study found berberine significantly inhibited cancer stem cell self-renewal and reduced tumor sphere formation in breast cancer models. Multiple studies confirm anti-proliferative effects in colon, lung, ovarian, and cervical cancer cell lines. A clinical trial found berberine supplementation significantly reduced the recurrence of colorectal adenomas, precancerous polyps, over a 2-year follow-up period. The traditional use: berberine-containing plant preparations consumed as decoctions. Available today as standardized berberine extract. The ancient compound that precedes modern pharmacology by millennia and targets the cancer cell population that modern pharmacology is still trying to eliminate. 16. Wormwood/Artemisia. The biblical bitter plant with an Artemisinin discovery. Wormwood appears in the Bible specifically as a plant of bitterness and consequence. Revelation 8 verse 11. Lamentations 3 verse 15. And in ancient medicine, the Artemisia family, including wormwood, was used across the Near East and Mediterranean for what healers described as abnormal heat in the body and unwanted growths. And here is what modern oncology has found. Artemisinin, extracted from Artemisia annua, sweet wormwood, closely related to the biblical plant, reacts with iron to produce free radicals specifically inside cancer cells. Cancer cells accumulate iron at dramatically higher concentrations than normal cells because iron is required for rapid cell division. When artemisinin encounters this concentrated iron in cancer cells, it produces a burst of free radicals that kill the cancer cell from within, while leaving iron-depleted normal cells largely unaffected. A study published in Life Sciences found artemisinin selectively killed cancer cells at rates 1,200 times higher than normal cells. Multiple clinical studies confirm activity against breast, prostate, and colorectal cancer cell lines. Research programs at the University of Washington and Johns Hopkins are actively developing artemisinin derivatives as cancer treatments. The traditional use: Artemisia preparations consumed as bitter tea across the ancient Near Eastern world. The bitterness the biblical world associated with consequence and purification was pharmacologically active in ways that oncological research is now actively developing into treatment protocols. 17. Elderberry, the dark berry that activates immune cancer surveillance. Sambucus nigra. Elder appears in ancient European and Near Eastern traditions as a sacred and medicinal tree. And for cancer prevention specifically, elderberry works through a mechanism that places it in a category distinct from every other plant on this list. And here is what modern immunology has found. Anthocyanins and specific polysaccharides in elderberry stimulate natural killer cell activity, the immune cells responsible for identifying and eliminating cancer cells before they establish as tumors. Natural killer cells patrol the body continuously. When their activity is high, precancerous cells are identified and destroyed. When NK cell activity is low, cancer cells escape surveillance and establish. A clinical study found elderberry extract significantly increased natural killer cell activity in healthy adults after 8 days of consumption. A separate study confirmed elderberry compounds significantly inhibited the proliferation of human colon cancer cells through combined direct, anti-proliferative, and immune-stimulating mechanisms. The traditional use: elderberry consumed as syrup, tea, or whole-cooked fruit. The ancient world that consumed elder preparations regularly was maintaining the immune surveillance that keeps cancer from establishing in the first place. 18. Cat's claw, the Amazonian vine that repairs DNA. Uncaria tomentosa, native to the Amazon. But the mechanism that makes cat's claw relevant to cancer prevention is DNA repair, and DNA repair is universal. And here is what modern oncology has found. Oxindole alkaloids in cat's claw inhibit NF-κB, which we have encountered multiple times on this list as a cancer-promoting pathway. But cat's claw's most distinctive property for cancer prevention involves its effect on DNA repair enzymes. Quinovic acid glycosides in cat's claw activate the nucleotide excision repair pathway, the cellular mechanism responsible for removing the bulky DNA lesions created by environmental carcinogens, UV radiation, and chemotherapy-related DNA damage. A study published in Phytomedicine found cat's claw extract significantly enhanced DNA repair capacity in human cells exposed to carcinogens. A separate study found cat's claw significantly reduced the formation of micronuclei, a marker of DNA damage and cancer risk, in patients exposed to DNA-damaging agents. The traditional use: cat's claw bark decoction consumed as daily tea, available as standardized bark extract. The ancient Americas independently discovered the same principle that modern oncological genetics is now studying in cancer prevention research. 19. Frankincense, the sacred resin that the Baylor study confirmed. Here we are at number 19. The anointing resin, the one the Magi carried. The Baylor University Medical Center study. Leviticus 2. Frankincense was commanded as part of the grain offerings in the temple. It anointed every sacred object in Israelite worship. And modern oncological research at Baylor University Medical Center published a study that brought frankincense back to the attention of cancer biology. And here's what the research found. Boswellic acids and frankincense, specifically AKBA, inhibit the LOXL2 enzyme, a protein associated with tumor invasion and metastasis. They also inhibit topoisomerase, the enzyme cancer cells require to replicate DNA during division, through a mechanism distinct from conventional chemotherapy drugs. And they specifically target cancer cells over normal cells because cancer cells express the target proteins at much higher levels. The Baylor study found frankincense essential oil significantly inhibited the proliferation of bladder cancer cells without affecting normal bladder cells. A separate clinical observation found boswellic acids significantly reduce cerebral edema in patients with brain tumors during radiation therapy, a finding so consistent that Boswellia is now used clinically in Germany for this purpose. The traditional use: frankincense resin consumed internally as a small piece dissolved in warm water, applied topically in oil preparations. The sacred anointing resin of the biblical world was delivering anti-cancer compounds through every surface it touched. 20. The ancient food practice, the pattern that made every plant on this list work. And here we are at number 20. Not a single plant. The practice that connected every plant on this list into the system that the ancient world lived without naming it, and that modern cancer epidemiology has now studied with enough precision to quantify its effect. Ezekiel 47 verse 12. Their fruit will be for food, and their leaves for healing. The ancient world did not separate the healing plant from the daily meal. The medicine was the food. The food was the medicine. Every meal was a delivery of the compounds that protected the body from the inside out. Modern cancer epidemiology has confirmed what Ezekiel described. The WCRF, World Cancer Research Fund, Continuous Update Project, which is the largest systematic review of diet and cancer risk ever conducted, has identified one dietary pattern more consistently protective against cancer than any other. Not a supplement, not a drug, a pattern. Abundant plant diversity, whole plant foods at every meal. Herbs and spices consumed generously as food rather than seasonings. Fermented foods, limited processed food, and red meat. That is the biblical dietary pattern. That is the diet of the Levant, of the Mediterranean, of every ancient population that lived alongside these plants and consumed them daily as a matter of ordinary life. They did not have a cancer prevention protocol. They had a table, and the table was doing what the protocol is now trying to replicate. The plants on this list are available. The pattern that makes them work is teachable. And the most important thing the ancient world understood about cancer, before they had a word for it, was the same thing the WCRF has now confirmed with 50 years of accumulated research. The food you eat every day, at every meal, for your entire life, It is either building the body's capacity to find and destroy abnormal cells before they become cancer, or it is not. The biblical world built that capacity into every meal. You can too. Drop a comment and tell me which plant on this list you are adding to your daily meals. Are you team turmeric and black pepper at every meal? Team pomegranate every morning for the angiogenesis